Hello, this is Dr. Vijay. Today we are going to study about beta oxidation pathway. In case you are new to this channel, please watch these videos in order to understand today's video. So in the first video I have explained about introduction. In the second video, explanation about lipolysis. Third video about activation of fatty acid in the mitochondria. And fourth video explains about carnitine shuttle transport system, and just previously uploaded video which explains about an overview of fatty acid oxidation, that is beta oxidation pathway. So far we have learnt that beta oxidation takes place in mitochondrial matrix. So now we have activated fatty acid, that is acyl-CoA, in mitochondrial matrix. So this is a fatty acid where this CH2 or hydrocarbon side chain will continue. So I have mentioned here R, that means it has got many hydrocarbon side chains. So this fatty acid oxidation I am going to explain for saturated and even number fatty acid. For odd number fatty acid and unsaturated fatty acid oxidation, that is a different story. I am going to make separate videos. Now we have activated fatty acid inside the mitochondrial matrix. The first reaction catalyzed by oxidoreductases or class 1 enzyme. Here in this case it is dehydrogenase. We call it as acyl-CoA dehydrogenase. Suppose this fatty acid is palmitic acid, then palmitoyl-CoA dehydrogenase. This fatty acid must undergo oxidation. That means this reaction should be coupled with the reduction reaction. The oxidizing agent going to be a coenzyme derived from riboflavin, that is FAD. So this FAD is in oxidized form. It will accept two hydrogen atoms from this fatty acid, from the second carbon atom and third carbon atom, or alpha and beta carbon atom. In turn, this FAD undergoes reduction, or it will reduce to FADH2, catalyzed by acyl-CoA dehydrogenase. Now if you remove two hydrogen from this second and third carbon atom of this fatty acid, the resultant intermediate would be like this. Now you can see there is an introduction of double bond here, and this CH2 and CH2 will become CH and CH. So the name of this compound is called delta 2 trans-enoyl CoA. So this delta means the numbering. Carbon number 1, carbon number 2, carbon number 3. If you want to study the numbering or naming of fatty acid, please go back to fatty acids video under lipid chemistry playlist. Now you can see there is a double bond here between alpha and beta carbon atom. Now this delta 2 trans-enoyl CoA undergoes saturation by adding water. Now the double bond becomes single bond here. You can see here. So water contains two hydrogen atom and one oxygen atom. This CH will become CH2, and here CH-OH group will be attached. So double bond becomes single bond. Now this intermediate we can call it as carbon number 1, carbon number 2, carbon number 3. So 3. And in the carbon number 3 we have hydroxyl group. So 3 hydroxy acyl CoA. And some textbook mentioned it as instead of 3 you can write beta also, because this is alpha carbon atom, this is beta carbon atom. You can call it as beta hydroxy acyl CoA or 3 hydroxy acyl CoA. And this particular reaction catalyzed by, since we are adding water, the enzyme is delta 2 enoyl CoA hydratase. The next reaction, this 3 hydroxy acyl CoA again undergoes oxidation reduction reaction. That means This 3-hydroxyacyl-CoA again catalyzed by a oxidoreductase, it is called 3-hydroxyacyl-CoA dehydrogenase. One more thing you need to remember, wherever there is enzyme dehydrogenase, it must contain either FAD or NAD. So here in this case, the coenzyme is NAD. This NAD is oxidized. It will accept 2 hydrogen now from this 3-hydroxyacyl-CoA. That means from the third carbon atom, it will remove 2 hydrogen, this hydrogen and this hydrogen, and this 2 hydrogen accepted by this NAD, in turn this NAD reduced to NADH plus H plus. Again this is a reducing equivalent like FADH2, and they will hand over their electron to components of respiratory chain. Later they will produce ATP. The resultant intermediate would be like this. It is called carbon number 1, carbon number 2, carbon number 3. 3, and we have removed 2 hydrogens. So this C double bond O is now keto group. So we call it as 3-ketoacyl-CoA, catalyzed by 3-hydroxyacyl-CoA dehydrogenase. So there are 2 oxidation reaction here. So this reaction is also oxidation and this also oxidation reaction. The next reaction, you can see here now, the 2 carbon unit, it looks like acetyl-CoA. This is carbon number 1, carbon number 2, carbon number 3, and this is alpha carbon atom and this is beta carbon atom. Now there is going to be cleavage or bond breakage here between alpha and beta carbon atom. So these 2 carbon units released by an enzyme called thiolase, which actually breaks the bond between alpha and beta carbon atom. So when these 2 units are released, the remaining will be again acyl-CoA, and we should add here coenzyme A. By addition of coenzyme A, there is going to be release of acetyl-CoA. Plus we have fatty acid or acyl-CoA. So this keto group now becomes carboxyl group of the acyl-CoA, and this coenzyme A is added here. Suppose this fatty acid contains n number of carbons, and the resultant fatty acid or acyl-CoA would be shortage of 2 carbon units, because this entire acyl-CoA we release 2 carbon as acetyl-CoA. So this pathway proceeds in a cyclical fashion, which results in degradation of these long chain fatty acids to acetyl-CoA. So suppose this fatty acid contains n number of carbon atoms. So this cycle n divided by 2 minus 1 cycle. Suppose this is a palmitic acid, it contains 16 carbon atoms. So 16 by 2 means 8 minus 1 means 7. There is going to be 7 cycles of beta oxidation for palmitic acid. This acetyl-CoA now enters TCA cycle. It will give ATP, carbon dioxide and water. And we have reducing equivalents here, FADH2 and NADH. They all enter electron transport chain and give ATP. Remember, one FADH2 after entering electron transport chain will give 1.5 ATP, whereas one NADH will give 2.5 ATP, which I am going to explain in the next video, that is energetics of beta oxidation. I will take a palmitic acid as a specific example, and we will calculate exactly how many ATPs are generated after complete oxidation of one palmitic acid. So this is beta oxidation pathway. You should remember, in beta oxidation there is going to be oxidation reaction here by FAD coupled acyl-CoA dehydrogenase, And the second reaction here, there is a hydrolysis by anyl-CoA hydratase. And the third reaction here, again oxidation by NAD coupled hydroxyacyl-CoA dehydrogenase. And the last reaction is going to be cleavage of this 3-keto-acyl-CoA into one molecule of acetyl-CoA and N-2 acyl-CoA, and this cycle continues until the entire fatty acid converted to acetyl-CoA. So this acetyl-CoA enters citric acid cycle to give ATP, carbon dioxide and water, and through electron transport chain. You should also remember, in this pathway large quantities of reducing equivalents like FADH2 and NADH are generated and are used to form ATP by oxidative phosphorylation. So if this is a palmitic acid, we will have 8 acetyl-CoA and 7 FADH2 and 7 NADH. So this is with respect to beta oxidation pathway, especially for saturated even number long chain fatty acids. Thanks for watching.